Hi, my name is Abby, and in today's video, we will look into the genetics behind DNA fingerprinting. We will begin at the scene of a murder. The victim is a man named Don Lewis. The responding officers at the scene found hair samples that could be from our murderer. To try to determine the hair's origin, forensic scientists will run a DNA fingerprint test. DNA fingerprint tests utilize aspects of a genome known as VNTRs, or variable number tandem repeats. STRs, or short tandem repeats, are sometimes used as well. VNTRs are sections of DNA that have tandem repeats of base pairs and can vary in length. These loci are highly variable between individuals, which allows us to use them for identification. A person will receive an allele from each parent, which means an individual will have one common allele for each VNTR locus with a parent. Because of this, we can figure out parental relations. Once the DNA evidence is found, it is put through a polymerase chain reaction, or PCR, to amplify the DNA. After PCR, the DNA is run through gel electrophoresis, which allows scientists to analyze the genetic associations. The police have 4 suspects for the crime: Don Lewis's ex-wife Carol, his co-worker Joe, Carol's current husband Howard, and Carol's daughter Jamie. DNA samples have been taken from each suspect, and a VNTR loci test was run. Here are the results. The victim's DNA was run to rule it out. On the first gel, it is evidence that his DNA does not match the crime scene DNA, as the bands do not line up. The second gel is consistent with the assumption, as the DNA bands also do not match. We can safely conclude that the hair was not from the victim. Moving on, we can see that Joe's DNA matches the recovered DNA at the first locus, as both bands match with the ones from the crime scene. However, looking at the second locus, the bands do not match up with the evidence, meaning that we can exclude him from the search. We need both loci to match for a conviction. Next, we will look at Howard. Howard's DNA bands match with the evidence left at the crime scene on the second VNTR locus. For the first locus, there isn't a match. Because Howard's DNA bands do not match on both loci to the evidence, we can remove him from suspicion. Jamie is next. As we can see from both gels, only one of her bands at each locus lines up with the DNA from the crime scene. She is not a match even with the overlapping bands, and therefore we can exclude her. But looking further into Jamie's bands, we can tell that Carol is her mother, as she has one allele in relation to Carol at both loci. We will look at Carol next. In the first gel, Carol's DNA bands match the ones from the crime scene perfectly. Looking at the second VNTR locus test to confirm, we can see a match between Carol's DNA and the evidence left at the crime scene. Carol's DNA bands are at the same locations as the crime scene DNA, making her consistent with the evidence. For the DNA fingerprint test, Carol could be the culprit from our murder mystery.